Hello and welcome, friends. Today we are going to discuss the next practical, that is methods of plant disease measurement. In that practical we discuss about how we measure the incidence of disease with the help of different methods. So let us see why the measurement of disease is essential. So as we know that if you measure the intensity of disease, so you should know that the prevalence of disease, that is, prevalence means the specific pathogen that cause specific damage to the specific host within a particular period of time or within a specific period of time. So you should know the list of such a pathogen that cause damage to the host crop within a specific period of time. Then you should know that the extent of damage caused by the disease. That means once the incidence of disease is occur on the host, then what percent of damage is caused by that disease. So measurement provides that important note. The next, if you know the nature of damage or if you measure the intensity of that disease, so we can take the suitable management practices. That means depending on type of disease, we can use chemical or physical or biological practice for control the spread of pathogen, or for control the incidence of pathogen, or before the incidence of pathogen we can take the some precautionary measures. So that it is very essential for the measurement of plant disease. Then plant disease measurement, it provides the information about what are the different hosts or the plant that shows a resistance against pathogen. That means if during measurement, if the intensity of pathogen is less, then that shows the resistance ability of, or that host shows a resistance ability against the host or against the pathogen. Next we discuss about what are the categories of damages. So in that, suppose if the incidence of pathogen is caused on the plant, sometime whole plant it get affected or destroyed due to the incidence of particular pathogen, and sometime only part of the plant or the part of the field is infected. For example, part of the plant, some of the leaves or tillers or stem, branches or fruits that are affected, and in case of whole plant, along with all parts of the plants, it completely damaged. So they, depending on the part of plant affected, it is categorized into two categories, that is whole plant infected, or the plant or the part of plant is infected from the field. The next, then here we discuss there are two types of the symptoms known as local symptoms or localized symptoms, and another is called as an systemic symptoms. So local symptoms, that means the symptoms around the site of inoculation are denoted. That is, here it is indicated here, once there is an incidence of pathogen, so at the point of inoculation only symptoms are occurred around the infection or around the inoculation, that shows local symptoms. But sometime some diseases, after causing local symptoms, that spread within a plant parts or within a different part of the plant. For example, they are moved through the stem, leaves or different veins, and it causes a damage or it cause a damage to the different parts of the plants. by spreading. So it is called as an systemic symptoms. That indicates here when pathogens spread from site of inoculation and cause symptoms in other parts of the plant. This is referred as systemic symptoms. So that we already described that. Suppose infection start from the roots and it will be move after causing infection here, it will be move through the system of the plant and causes symptoms around a different plants or around the different parts of the plant. Then it is called as an systemic symptoms. That means it move through the system of the plants. So this is about localized symptoms and systemic symptoms. So again we discuss here localized symptoms. That means when the inoculation of pathogen is takes place or insertion of pathogen is takes place and symptom is caused around the infection, it is called local symptoms. And when the pathogen is spread through the system of the plant and cause symptoms on different parts of the plants, then it is called as systemic symptoms. Next we discuss here the types of diseases. So depending on the different parameters, the diseases are categorized into two diseases or different two types, that is simple interest disease and compound interest disease. So simple interest disease, that means what? Those pathogen which complete only one life cycle in their single season, then it is called as a simple interest disease. In contrast with that simple interest disease, what is meant by compound interest disease? Compound interest disease is nothing but those pathogen which complete several or many life cycles in a one season, then it is called as an compound interest disease. So example of simple interest disease, that is most of the soil borne diseases or pathogen it comes under simple interest disease. For example here for example wilt, then smut, for example loose smut of wheat, then different kinds of rot, damping off, they comes under simple interest disease because that pathogen complete only one life cycle in their one season. And the example of compound interest disease is blight, leaf spot, rust, for example wheat rust, which is caused by Puccinia graminis. So they comes under compound interest disease because it takes several or it completes several life cycle in their one crop season. Then in simple interest disease, that pathogen requires or the incubation period of such a disease is long. That means more time is required for the development of for the spread of pathogens. And in case of compound interest disease, incubation period is short, that is within a short period of time symptoms develop rapidly and the spread of such symptoms or pathogen is takes place. Next one, the diseases which comes under simple interest. So such a diseases or the pathogen are transferred only to a short distance with the help of, for example, water. In case of compound interest disease, the pathogen they transferred to a long distance with the help of, for example, air or wind. Here example, here it is mentioned all the examples, that is simple interest disease and the compound interest disease. Here SID, that is simple interest disease, compound interest disease. Example is given: damping of, wilt, smut, burnt, fruit rot, virus diseases. And in case of compound interest disease, rust, spot, blight, blast, powdery mildew and downy mildew. So they comes under compound interest disease. The next one is there, plant disease are measured as a disease incidence and intensity or disease severity. So let us see what is meant by disease incidence and what is meant by disease intensity or severity, or how the incidence is calculated, or how the disease intensity or severity is calculated. So let us see what is meant by incidence. It is nothing but the proportion or percentage of diseased part or leaves, shoots, tillers or ear heads, grains, seedlings or plant parts. That means in case of percent disease incidence, we count the total number of diseased plants and we count the total number of plants observed. And when we divide and multiply it by 100, then it gives the percent incidence of particular disease. For example, total number of plants observed, that is 10, and out of 10, suppose 5 plants is observed or diseased plant is observed, so into 100, so it gives a 50% incidence of that disease here. So in that way we have to calculate the percent incidence of disease of particular plant. So this is about your percent incidence of disease. So in that percent disease incidence, we have to made the different grades according to the incidence. Suppose 0 to 0 or 0% disease incidence is there, that means your crop is completely free from microorganism. Suppose disease incidence is occur from 0 to 5, so your host or plant is resistance. Suppose PDI, that is percent disease incidence, is 5 to 15, so your crop is moderately resistance. Then 15 to 13, here 15 to 30, that is moderately susceptible, and 30 to 50, your crop is susceptible. And if the PDI is above 50%, so it shows your crop is highly susceptible to the pest or disease. So that indicates, that whole indicates the, that PDI or percent disease incidence, that indicates the grades. That means it is a free, resistance, moderately resistant, susceptible, highly susceptible, etc. Next we discuss our that incidence or that percent disease incidence. It is generally done the disease like the damping of wilt, smut, bunt, fruit rots and different viral diseases. In that disease we use the percent disease incidence. Next, the intensity or severity of disease. So let us see what is meant by intensity or severity of disease. Here the disease severity is the percentage of relevant host tissue or organ covered by symptoms or lesion or damaged by the disease. In that, the disease severity results from the number and size of the lesions or the spots. So in that we discuss the percentage of relevant host tissue or organ. That means what? Suppose that size will be there or spot will be there. So depending on the size of the spot which is covered on host, that intensity is calculated. For example, depending on the size of the spot or the number of the lesions or the spots, we have to conclude that at what percent that disease intensity is occurred. Here that picture indicates only 1% disease incidence is occurred on first leaves. Here near about 5% disease incidence is occurred on second leaves. Here 15% and here 25% disease incidence. That indicates as the number of spot and the size of spot increases, then intensity of disease is also increases. Here in second picture, second, here same thing is happens, that is as the size and the number of spot is increases from first, second, third and fourth, the disease intensity is also increases. That means depending on the number of lesions and size of the lesions, intensity of disease is increases and it is also measured in a percentage. So in next example we discuss about how we have to calculate the disease severity. So plant disease severity or percent disease index is calculated by using the formula, that is PDI is equal to summation of numerical ratings divided by total number of leaves observed into maximum grade into 100. When we discuss about when we solve the examples based on PDI, so at that time we discuss about the meaning of that numerical ratings or total number of leaves observed or what is maximum grade. So that formula was given by the scientist McKinney in 1923. For calculation of standard or for calculation of plant disease intensity, we have to use different visual assessment methods. In case of that, we use two methods, that is standard area diagram and descriptive keys for the assessment of plant disease intensity. So in standard area diagram, here it represent the different percentage of disease severity level, or standard area diagram provides the information about how much plant part is affected or how much the disease will be occurred on a particular leaves. For example, that standard area diagram, that is example leaf rust of wheat, provides 1, 5, 10, 20, 50% leaf area affected. Here just like this 1%, 5%, 50% or 25%. So depending on the size or the number of lesions, the disease severity is estimated with the help of such a standard area diagram. So this shows the standard area diagram that indicates the percent of host area is affected by the disease. So that second image also provide the estimation of disease intensity. It is less severity, that is 10%. In case of second, another severe incidence of disease, and in case of that 50% and above, that is that crop is affected by or wheat is affected by more severe disease, that is a septoria. Next we discuss about the descriptive skills. So that descriptive skills, the information about disease intensity in different grades, that is in different numerical scales or depending on the disease rating. So let us see one by one. First, here scale is ranges from 0, 1, 3, 5, 7 and 9. That means here it indicates 0 to 9 scale. That is here 0 to 9 grade disease scale is indicated. That is 0, that means leaf area free from infection. That is here it indicates leaf area is free from infection, that is 0. Second one, first, or the grade is first, leaf area infected up to 1%, that is it indicates leaf area infected up to only 1%. Next, here third, leaf area infected up to 10%, that is if you see the third grade, that is only infection takes place but up to 10%. In fifth, leaf area infected up to 25%, that is in fifth picture shows that 50% area is infected by the disease. Then in 7, leaf area infected up to 50%, that is severe damage, that is 7 here. Leaf area is infected up to a 50%. And in case of 9, leaf area is infected, that is more than 50%, that is very severe damage takes place due to the incidence of disease. So with the help of different grades, the infection or the amount of infection is indicated. For example, here again we discuss here about. So let us see the example. Here 0 to 9 grade is mentioned. That is, as we previously discussed about 0 to 9 grade. What is 0 to 9 grade? That 0 means no incidence, 1 means only 5%, 3 means only here only 10%. So in that way, as we know that the 0, 1, 3, 5, 7, 9 grade is mentioned. So we have to observe the here total ratings. That means total number of leaves observed. So when we observed 5 leaves, they comes under 0 grade, that is no incidence of disease. Next 5 leaves are observed, they comes under 1 grade, that is disease incidence. That is on 5 leaves only 1% disease incidence is occurred. Then 8 leaves indicates 3% disease grade. That means what? Here 3%, that is 10% disease incidence is takes place. Then 4 leaves indicate they comes under 5. Here 5, that means they indicates 25% disease incidence is occurred. Then 7 here 8 leaves indicates, or 8 leaves is infected, and they comes under the 7 grade. That means near about 50% disease incidence is occurred. And in at the fourth or 4 leaves is assessed, they comes under 9, because near about more than 50% disease incidence is occurred. So this is called as a total rating. That means total number of leaves is assessed or observed, and they categorize into a different grades according to the severity of that particular disease. Yeah. Then in that grade, just we have to multiply, that is disease grade into total rating. That is, if you want to obtain, if you want to know the number of ratings, so simply you have to do the 0 into 5. 0 into 5, so it will give 0. It will give 0. Then here 1 into 5, this is grade, that is 0, this is grade 1 and total rating 5, that is 1 into 5, that will give 5. Then 3 into 8, that this is grade 3, total rating 8, so 24, that is it indicates 24. Then 5 into 4, that is 20. 7 into 8, 56. 9 into 4, 36. So this is the number of ratings. This is a number of ratings. So when we do the summation of number of ratings, so near about 186 number of ratings are obtained, and here total number of ratings is 34. So depending on the or based on the formula of disease severity, that is, as we know that previously, you should know the formula, that is here the numerical, that is PDI is equal to, or percent disease incidence or index is equal to summation of numerical ratings divided by total number of leaves observed into maximum grade into 100. So according to this formula, you should know the here the summation of total ratings that will given here, that just like this 186, okay, divided by total number of leaves is assessed, that is total rating, that is 34, that is 186 divided by 34 into maximum grade. Formula is maximum grade, so maximum grade is here 9, so into 9, and it is in expressed in percentage, so into 100, so this will be, disease severity is 60%. So in that way we have to calculate the disease severity. It is a very simple for calculation. So again we discuss disease grade, that is 1 to 9 or 1 to 12, depending on the host we have to calculate, or already given total rating, that means number of leaves observed or assessed for the presence or absence of disease, and here number of ratings that will be obtained by multiplying disease grade into total ratings, and depending on the formula we have to calculate the disease severity. So in that way we have to calculate disease severity. So if you want to more information about disease severity and disease intensity, so please refer that book, that is Phytopathometry by V.V. Datar and C.D. Mai. Students, I hope you will understand it was a simple topic. If you have any difficulty regarding that topic, so please ask me on my WhatsApp number. Thank you.